All right. The next speaker is Martha Sim. She will be talking to us about spatial dynamics in blood clotting regulation.
Testing. Hello, everyone. You might have heard that blood is thicker than water, and this ancient wisdom holds more truths in it, as it can also be said that blood can more easily or has a great tendency to coagulate or to clot, which is when it turns into this more gel-like consistency. And this is important as during evolution, human needs to be able to stop bleeding very quickly and very efficiently as we were trying to survive in the wilderness amidst the, predators in the primitive world. However, with this greater tendency for clotting comes a greater price in how excessive clot can also form within intact blood flow. And this can lead to heart attacks and stroke, which are the leading causes of death in the modern world as our lifestyle is becoming more sedentary and our environment becoming more safe physically. And so the proteins in the blood that promote clotting are called procoagulant proteins are not only very powerful, they're also very abundant, whereas the anticoagulant proteins are very spare. And they're also very ineffective when studied in a test tube in the lab environment. Whereas we have a lot of drugs that are effective, very effective in treating clotting, every single one of them would cause bleeding as well as a side effect. And so our lab studies coagulation and anticoagulant proteins in how they function in preventing excessive clotting. And we also aim to separate this effect within normal injury response versus excessive clot formation. And so in order to do this, we collect blood and platelets, which are the cells most responsible for forming clot. And then we study them, with various proteins, which also means that I will, from time to time, need to walk around the lab looking for victims, kind donors to give me blood so that I can study them. But we also grow cells that make up the blood vessel wall. And then we study the anticoagulant property. And what we found was that within the clot, the platelet surface serves as this, promote this procoagulant tendencies in which anticoagulant proteins inside this are almost ineffective completely. Whereas inside the bloodstream, the circulating cells and the cells that make up the blood vessel walls promote anticoagulation efficiency. And so this serves as this spatial dynamics in controlling clotting, in which we have checks and balances to prevent excessive clot from forming. And also, finally, this can also serve, to help us develop better drugs to treat clotting better while maintaining or preventing bleeding as a side effect. Thank you for your attention.